Assalamualaikum and hello everyone. So today we going to present about genetically modified banana. So my name is Ridwan Salim. So this is our group members. So first of all, as you can see, there is main factors contribute the development of genetically modified banana. So this point I get it from Wang et al. 2021. So the first point is the balance between crop production and disease resistance is a major challenge facing modern agriculture, which means crop production and disease resistance is a problem that can contribute the development of genetically modified banana. So number two is there is no resistance to fusarium wilts, which cause plant damage and eventually death. So number three is we want to achieve the ideal plants architecture, which is IPA, because these banana varieties are desirable because they have high logging resistance, optimal photosynthesis, and efficient water absorption. And the last one is banana production is low.
Before I move on to how transgenic banana is developed, we must first know why transgenic banana is developed. Okay, the causal for transgenic banana is developed is because of the disease called FOC. FOC is a short-term stand for Fusarium oxysporum cubensi. It is one of the deadly crop species and cannot be controlled by the means of biological, chemicals, or even physical controls. So under FOC, there are 4 types of races, which are race 1, 2, 3, and 4. And each of these races give different effects to the host plant. Deadliest of FOC disease is actually under race 4, which we call as tropical race 4 or TR4. So TR4 causes the banana crops to be discolored. Then necrosis in roots and stem, which continue to be wilt and die. So there is no effective control to stop TR4 since it is a soil-borne disease, but we can develop banana which cause to resistant to TR4. So how transgenic banana is made? First, it used the technique of centrifugation-assisted agrobacterium-mediated transformation to transform the immature male flowers into embryogenic cell suspension. It used 2 types of binary vectors, which are RGA2 and also CED9, and these 2 are tested on Musa acuminata Cavendish. Following the transformation, the embryo is selected and regenerated and screened by using PCR. So the results from the PCR, there are about 5 lines from RGA2, CED9, and also controls are selected and analyzed again using southern blots or southern analysis. So the last step is the field testing, where all the 5 lines of control, RGA2 and also CED9 are tested on a plot. And the result is that it is proven that CED9 and also RGA2 are resistant to TR4. Thus, the banana crops subspecies Cavendish is commercialized for its ability to resist TR4 disease.
Now, we move on to the next slides. The question is, do the transgenic crops help in reducing the limitation on the white banana? So the answer is yes. Here are the reasons. Fusarium wilt is caused by a fungus that lives in the soil known as fork. Fork is the most serious threat to banana production because it will completely destroy the plants once it enters into the plant, and it is unfortunately cannot be controlled by any chemical or physical means. As for example, when the banana tree was infected with TR4, which is one of the distinct races of fork, it will show several symptoms such as discoloration, followed by necrosis in the root and rhizome that prevents water and nutrients from being transported, resulting in withering and eventually death of the entire plant. So in order to prevent from infected of fork, banana plant needs to undergo genetic modification that aim to produce plant that resistant to fork, as most of cultivars banana are sterile. Thus, banana plant that undergo gene modification for fog resistance will successfully grow without being infected by Fusarium wilt. So, this genetic modification is an effective method of developing bananas with improved agronomic traits such as increased disease resistance and also yield.
Next slide is about positive and negative impact of genetic engineering. For the positive impact, first, it is one of the premium methods for developing banana with improved agronomic features. Second, it also can create genetically altered elite banana cultivar with superior disease resistance, optimal architecture, and can also produce higher yield. Next, if there is enhanced knowledge about banana-specific pathogenic and infectious mechanism, so this mechanism can allow development of disease-resistant banana by using siRNA targeting specific key pathogenic genes, and also can use the HIGS system. Next is the negative impact. First, due to banana's genetic sterility, the forward genetic approach, such as genetic segregation tests, make it difficult to find potential genes that can associate it with desirable characteristics. Next, the method that are created is dependent on type of the situation, which means they need urgently stable transformation and regeneration protocol that are applicable to all banana varieties.
The last one, we go to the ethical issues. So the first point is uncertainty of the safety of humans after consumption. So first, the allowance of the free use of GMOs in agriculture is associated with the unknown potential health effects of consuming chemically treated foods. So actually, customers assume that the foods they buy are safe, even though many contains compounds that are potentially toxic and or allergics. So number two is uncertainty of the safety of the environment. So some of these issues include the emerge of superweeds and superbugs as a result of antibiotic resistance to GM, pesticide and chemicals.